All right, next up we're going to talk about the form and function of the male reproductive system. The male reproductive system is going to consist of several organs, starting with the testes. Then we're going to look at the duct system, which will include the epididymis, van deferens, and urethra. And then we'll look at the accessory glands, the prostate glands, seminal vesicles, bulbourethral glands, and then finally the penis. So figure 17.1 is a fantastic summary slide. Really it is. It'll show you pretty much everything you need to know. By the end of this chapter, you should be able to cover up all of the words on either side or all of the text on either side and just walk yourself through all of the major organs of the male reproductive system. But we're going to start off by breaking each one of these down individually. We're going to be looking at the testes. The testes are going to be, two of them, they're going to be held outside of the abdominal cavity inside of the scrotum. Why is this? It's cooler. It allows for a lower lower temperature, which is going to be important in the production of sperm. The sperm are going to develop in the seminiferous tubules right after puberty. Interstitial cells of the testes are going to produce androgens, including testosterone. These are going to be hormones that are going to help with the development of secondary sex male characteristics. A homeostatic imbalance of the testes may be testicular cancer. It's going to be the most common form of cancer among males 15 to 35 years of age. It usually does not cause pain, so monthly self-examinations are very important, as this might be the only way to detect it. There is a very high cure rate when it is caught early, so this is another reason why monthly self-examinations are so important. Moving on to the duct system. The duct system is going to contain several parts: the epididymis, vas deferens, and urethra. The epididymis is going to receive the sperm from seminiferous tubules. This is going to be the site of sperm maturation and storage. The vas deferens is responsible for conducting sperm from the epididymis to the urethra. The urethra is going to conduct urine from the urinary bladder. It also conducts sperm from the vas deferens. These are tubes that the sperm are going to travel in after being made in the testes. Their primary function is going to be transportation, storage, and maturation of the sperm. Here is a diagram going through the different points of the testes, vas deferens, and epididymis. Next, we're going to look at the accessory glands. The accessory glands include the prostate gland. The prostate gland is going to surround the upper portion of the urethra. This produces an alkaline secretion that activates sperm and reduces acidity of the female reproductive tract. There are two homeostatic imbalances that are related to the prostate gland. Both of them are going to be, one of them will be age-related enlargement. Age-related enlargement begins at middle age, and it may ultimately restrict flow of the urine. Cancer may also be found in the prostate gland. It can be detected through a rectal exam or through blood tests that measure prostate-specific antigens. The seminal vesicles are the next structure. These are going to be paired glands. They're going to secrete, the secretions found here are going to nourish the sperm, so they're going to be extremely high in fructose. They also help thicken the semen with the aid of amino acids and assist in the movement of sperm in the female reproductive tract. That would be the prostaglandins. The next accessory gland is going to be the bulbourethral glands. This is going to be another pair of glands. They're going to release a clear, slippery liquid before ejaculation. It may help to rinse acidic urine from the urethra and aid in the sperm's journey. All of these secretions are going to contribute to the fluid that makes up semen. Only a very small amount of semen, only about 5% of semen, is actually sperm cells. The rest is going to be a combination of these different fluids. Finally, the last organ that we're going to discuss is the penis. The penis its primary function is to deliver sperm to the female reproductive system. The glans penis is going to be the tip of the penis. It's full of many sensory nerve endings. This is going to be covered by a foreskin, which is sometimes surgically removed by circumcision. The penis is also going to contain 3 columns of spongy erectile tissue that are filled with blood during an erection. These three columns of spongy erectile tissue, they contain arteries arteries entering the penis are going to start to dilate. The erectile tissue is going to fill with blood. Veins draining blood from the penis are going to be squeezed shut so that the blood is trapped inside the penis. The blood is going to accumulate. The penis is going to swell. And this is what causes an erection. Homeostatic imbalance associated with the penis would be erectile dysfunction and or impotence. This is an inability to achieve or maintain an erection. The causes of these issues range from psychological problems to damaged nerves or blood vessels. There are certain medications that can prolong the effects of nitric oxide, which promotes the widening of arteries in the penis. Table 17.1 is a great place to look for a review of all of the different structures and functions of the male reproductive system. The last thing that we're going to talk about in the male reproductive system is going to be the sperm cells themselves. So here we're looking at we're looking at a sperm cell. The sperm cells are going to develop through a process known as spermatogenesis. This is going to occur within the seminiferous tubules. It's going to ultimately be responsible for reducing the number of chromosomes to one member of each pair. The changes in the shape of the sperm, or ultimately, once we reduce our chromosomal number, we're going to start to see during the process of maturation changes in the shape of sperm so they can deliver chromosomes. Some of the things that we are going to see during this process of maturation is going to be the development of three distinct regions. First region is going to be the head. The head is mostly just a nucleus with 23 chromosomes. On its tip, it has what is known as an acrosome. It's a vesicle with enzymes. Those enzymes are going to aid the sperm in penetrating the cells that are located around the egg. The next section is going to be the midpiece, this region found right in here. The midpiece has a lot of mitochondria, so just packed full of mitochondria. Why? Because of the giant long flagella. The mitochondria are going to provide ATP for the tail. The tail is going to be made up of a great big long flagellum. Its primary function is going to be motility. There are a couple of hormones that play a very important part in the male reproductive system. Testosterone is a main one. It's going to be secreted by the interstitial cells of the testes and is important for sperm production and the development of male secondary sex characteristics. Production is going to be regulated by a negative feedback loop. So the hypothalamus is going to release gonadotropin-releasing hormone. Gonadotropin-releasing hormone is going to stimulate the anterior pituitary to secrete luteinizing hormone. That luteinizing hormone stimulates the production of testosterone by the interstitial cells of the testes. Rising testosterone levels are going to inhibit the release of the gonadotropic releasing hormone, which is going to decrease the luteinizing hormone, which is going to decrease the testosterone. Decreased testosterone is going to increase gonadotropin releasing hormone. Gonadotropin releasing hormone is going to stimulate the luteinizing hormone. Luteinizing hormone is going to stimulate the release of testosterone, so on and so forth. So that's going to be that negative feedback loop. Sperm production is controlled by that negative feedback loop. And it's going to, because of that constant feedback loop, we're going to have that relatively stable levels of testosterone until later in life. So sperm production is controlled by that negative feedback loop, specifically by follicle-stimulating hormone, which is produced by the anterior pituitary that makes the cells that will become sperm more sensitive to testosterone. That will stimulate sperm production. High sperm numbers will then prompt the seminiferous tubules to produce an inhibitor, which will inhibit the production of gonadotropin-releasing hormone, and then thus decreasing the production of follicle-stimulating hormone. So again, this is going to function on that negative feedback loop that we've already mentioned.
The structures of the male reproductive system produce and store sperm, synthesize accessory fluids, and transport and deliver semen. Here we see a cross-section or side view of the male lower torso. The urinary bladder is a muscular sac that stores urine. It shares its exit tube, the urethra, with the male reproductive system. The urethra carries both urine from the bladder and sperm in the semen through the penis to the outside of the body. The erectile tissue of the penis is a spongy tissue surrounding the urethra that inflates with blood during sexual arousal and causes an erection. This mechanism provides for the delivery of semen into the female reproductive tract during ejaculation. Within the scrotal sac, or scrotum, are two paired testes, the site of spermatogenesis, and the source of male-specific hormones called androgens, the most important of which is testosterone. The newly formed sperm leave a testis via the epididymis, a coiled tube where the sperm continue to mature and can be stored until ejaculation. The sperm leave the epididymis via the vas deferens, which travels up and behind the urinary bladder and back down to the ejaculatory duct. Several accessory glands contribute accessory fluid to sperm as it proceeds. These are the seminal vesicles, the prostate gland, and the bulbourethral glands. Now consider each structure of the male reproductive tract in front view. Note that the vasa deferentia pass behind the urinary bladder. From this view, you can see that many of the structures, including the testes, epididymides, vasa deferentia, seminal vesicles, and bulbourethral glands, are paired. The cells that undergo spermatogenesis line the walls of the seminiferous tubules located in the testes. The parent cells are diploid and undergo mitosis to replenish themselves and to produce cells called primary spermatocytes. Meiosis I in the primary spermatocytes produces secondary spermatocytes. Meiosis II in the secondary spermatocytes produces haploid spermatids. The spermatids then undergo restructuring. Among other changes, the spermatids lose some of their cytoplasm and develop a tail. Mature sperm, also known as spermatozoa, are the result. During ejaculation, sperm are released into the female reproductive tract. If the sperm encounter an egg, they can come together in the process called fertilization. First, the sperm must move through the follicle cells that surround the egg. A covering called the zona pellucida provides the egg protection and acts as a species-specific barrier to fertilization. Sperm have receptors that recognize and bind to the zona pellucida from their own species. After binding, a sperm releases enzymes from a structure called the acrosome. Acrosomal enzymes break down the zona pellucida and allow the single sperm to contact the egg's plasma membrane and fuse with it. Reactions in the egg prevent additional sperm from entering. The DNA of the sperm unites with the DNA of the egg, forming a zygote. The zygote then begins the process of development and completes its journey with implantation into the uterine wall.